Yeah, so the tisotumab vedotin is a antibody-drug conjugate. The target is tissue factor, and the warhead is MMAE, so the auristatin analog, which is targets the microtubules. This is there are a fair number of ADCs that are in development, and I think that the kind of the concept is that it is a targeted way to deliver chemotherapy, taking advantage of factors that might be relevant for tumor biology. And in this case, it's tissue factor. And most people think, wait a minute, I remember tissue factor. That's a blood coagulation thing. And the answer to that is correct. It's factor 10, and tissue factor is responsible for the transition from factor 10 to factor 10A, which is a part of the extrinsic pathway. So when you get a cut, this is the kind of the factor that leads to a ultimate formation of a blood clot. So it just so happens that this particular mechanism is also hijacked by cancer cells and is responsible for a number of different components in the oncogenesis pathways, such as invasion and metastasis. And so the fact that it's expressed on a lot of cancer cells independent of normal tissue cells made it a very nice candidate for developing an antibody-drug conjugate. And so like most other antibody-drug conjugates, it uses tissue, it uses a target, which is in this case this tissue factor to deliver the warhead to the cancer cell, which gets internalized, enters into the lysosome, gets cleaved, and releases the warhead into the tumor microenvironment. So the nice thing about this particular compound is that because this particular the MMAE factor, we think that there's probably a significant bystander effect, so it gets released with unknown cell lysis. And there's also this concept that maybe it's also initiating an immunogenic cell death pathway with activation of ADCC and ADCP phagocytosis pathways that might make it a nice partner to an immunotherapy type strategy. Something that we would love to leverage even further. But that's kind of the basic nuts and bolts of the drug.